So we're going to go ahead and get started. Good morning and welcome to the Harmful Algal Bloom webinar series. My name is Laura Freskin and I'm from the Iowa Water Center located at Iowa State University. I will be your moderator today. The webinar is hosted by the Algal Bloom Action Team, of which Iowa State is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes, the North Central Region Water Network, and University Extension within each state of the North Central region. In addition to hosting a webinar series, our team is developing a website of resources, including fact sheets, and frequently asked questions on HABs topics. I encourage each of you to visit our website, which I will put in the chat in just a moment. There you can explore some of the resources we have to date, including a what you should know fact sheet, a HABs frequently asked questions database, and recordings from previous webinars, and our virtual HABs research symposium, which was held in January. Before we get started, there's going to be a couple housekeeping items. We will have a Q&A session after our speakers present today. Please list your questions for our speakers in the Q&A panel, and we will do our best to address all of them following the presentations. If there is a question already posted to the Q&A panel and you have the same question, upvote the question and we'll be sure to get answers to the most popular questions during the session. Today's webinar is being recorded and will be posted to the Algal Bloom Action Team website following today's session. If you're having any technical issues or have any questions about the Algal Bloom Action Team, please list those in the chat as well, and we'll be happy to assist you as best we can. Today's presentations focus on forecasting HABs and developmental impacts of neurotoxin exposure on aquatic populations. And we have two great presentations lined up for you today. Our first presentation will be brought to you by Dr. Richard Stumpf. Dr. Stumpf has over 30 years of experience in coastal oceanography with particular interest in harmful algal blooms. He develops methods to use satellite data and modeling to understand coastal eutrophication, habit change, and algal bloom monitoring and forecasting. He leads NOAA's efforts to translate forecasts of harmful algal blooms from research to operations. He received a BA degree in the environmental sciences from the University of Virginia and MS and PhD degrees from marine studies from the University of Delaware. So now we'll begin our first presentation. I will stop sharing my screen so you can share yours. Thank you very much, Dr. Stumpf.
Okay, thank you, Laura. Thank you for having me, and I guess I'll get right started in here. I assume you can see my screen okay? Okay, excellent. So I'm going to talk a bit about our monitoring and forecasting algal blooms. The monitoring is working primarily with satellite data in order to get, the intent is to get rapid synoptic views of blooms across the country. And our forecast, I'll be talking mostly about Lake Erie, but it provides a context for what could be done on other lakes around the country. Even though Lake Erie is a great lake, much larger than many, there's applicable characteristics to many lakes. So as many of you know, these blooms, cyanobacterial blooms, can produce extensive scums. An example here from Lake Erie. And whoops, I did something wrong here. Okay, still on screen. Okay, and satellite view of that same day shows that's just a small area in one of the islands of Lake Erie inside a much larger bright green bloom. Actually, Lake St. Clair to the north.
It looks like we stopped seeing your screen here.
Oh, that's not good. Let's try this again. How's that work?
There we go. That looks great. Thanks.
Okay. I hit a wrong button at the wrong time, so that caused that. So that bloom is in Lake Erie. You can see an extensive one in this area. This is well over 100 miles here, and Lake St. Clair in the north also had a large bloom at that time. But it gives you a sense of the scale that these blooms can approach. There are, of course, around the country, this is USGS data collected from the National Lake Assessment, showing presence of cyanobacteria around the country and lakes used for that. The red marks are at concentrations that would definitely pose a risk according to some of the World Health Organization, WHO standards. And while not all of these are toxic, many are. I should note that toxicity of these blooms varies, and in this case, only microcystin was looked at or presented here, but there are other toxins, anatoxin A and saxitoxin and cylindrospermopsin, all of which can be produced that can be problems in some of the other lakes. So this is a concern we need to consider from both perspectives. Satellite data, we're concentrating on Sentinel-3, which is out of the Copernicus program. It's a European satellite. And the sensor, the OLCI, which is Ocean Land Color Imager, OLCI, has bands that are suitable for detecting cyanobacteria and also estimating the concentration, particularly with chlorophyll. I put in a couple of examples. True color, and one of the points I want to make is people often, true color is great. It gives you an idea of what's going on, but you would have to squint very hard at this image of Wisconsin to try to see what's going on in Winnebago or in Green Bay. And some of these other lakes, you can barely tell this is greenish, but it's we're not looking at how green the water is. We're looking at spectral characteristics specific to cyanobacteria. Example from Florida also, and again, the same thing. There's a bloom in Lake Okeechobee you can't quite pick out. Same with some of these others. So we're able to pull these out. I'm not going to go into details of the algorithm. We have a number of papers on those. But the key thing is the sensor can pick up chlorophyll A quite specifically and also detect the presence of phycocyanin. And phycocyanin is a key indicator of pigment for many freshwater cyanobacteria. So we have that ability to pick it up, and I'll note how does that compare to field measurements. We compared the cell counts. The method was developed in Lake Erie, and cell counts from New England and Florida, and also Grand Lake, St. Mary's in Ohio, showed a close fit of what we determined from Lake Erie to these other areas for cell counts. And so we are definitely picking up the cyanobacteria. Microcystis, we call it microcystis equivalent cell counts, because we did this during a microcystis bloom, and other ones may vary somewhat, but you get similar results overall. And then the other question is, what about the chlorophyll A? Because that's really captures a biomass metric, and we're picking that up. And so a more recent study we've done looking at chlorophyll A in lakes that are tending to have cyanobacteria blooms, you can see we are picking up the chlorophyll A quite well. And this is examples from places around the country, a lot in Minnesota and Florida, but also Nevada, scattered ones in other regions. And this is pretty consistent on our ability to detect this. So we are able to pull this out with the satellite data quite well. One powerful characteristic of satellite is it, in some sense, is a virtual time machine. If you're doing water samples, if you didn't set up the sampling program or were able to preserve samples, you don't have the data. And with satellite, we started the methods actually in 2008, 2009 in Lake Erie, and we were able to go back in time. And we can also revise and improve them, which we've done. So there's a real advantage of this. This is Western Lake Erie. Detroit, this is the Detroit River up here. Toledo is over here, and Cleveland is in this area. And you can see some of the, also it's worth noting, some of the worst blooms look quite different. This one extended well across the lake. In contrast, in 2013, it was incredibly intense, but it stayed in it was in a very narrow area overall. And this can be true regardless of the size of the lake. This is a function of wind patterns. as well. And so if you tend to have in Lake Erie, it's somewhat of a prevailing westerly wind, you can get the bloom spinning eastward. If you have a northerly wind, it can end out on the US coast and to the relief of Canada. And if it's a southerly wind, it ends on the Canadian coast to the relief of the US, which it's not a great way to approach. And so we're able to look each year at the data. This is what the peak, this is the peak every year, and this is what 2022 looked like as well. It's not one of the worst years, but there was an intense bloom in the very southwest area of the lake, noticeably worse than 2021. And this is done off of 3 different satellites. We had to improvise with MODIS, but Maris and Olchi look quite similar. Olchi is now an operational sensor and the others are not. Key factor of cyanobacteria is phosphorus, and it's well established that excessive phosphorus does promote cyanobacteria blooms. This is an example of once you exceed about 100 micrograms per liter total phosphorus in the lake, pretty much you're high risk of having cyanobacteria being present. When you're at lower levels, it does not do as well. The cyanobacteria thrives best in high phosphorus environments, and diatoms and green algae tend to outcompete in lower phosphorus. Now, the type of phosphorus does matter. When you're in the lake, most of the phosphorus gets bound up in the cyanobacteria bloom. This is the actual inputs, in this case, river inputs to lake, and these are the concentrations. What I'd like to note is over this time period, there's pretty much no variation in the particulate phosphorus. And we went from pretty much no blooms in the 90s to a lot of blooms and starting about 2008 or so. And you can see a big shift in the dissolved reactive phosphorus, and that's the dissolved reactive phosphorus is most bioavailable, the phosphorus most accessible to the cyanos. And notice that it's actually up at around this 0.1 to 100 micrograms, just the dissolved phosphorus. And that's when the bloom conditions started to occur. We saw a near doubling of this. So this is a key factor. This concentration really does make a difference. And that does indicate there's a potential path out by reducing it again. There's the primary explanation for this has to do with some of the changes in farming practices involving no-till farming and tile drainage and the rest. And there's a long discussion. There's some very good papers explaining how this works, produced by USDA and others. Now, a key part of this is the timing of these loads, and it's the spring and early summer loads that really matter. That load during that time from March to June or July is what drives the bloom. It tends to stay in the lake, stay in the surface, probably the surface fluff layer, and then gets reused by the bloom. Some of the mild bloom years have followed, say, winters or falls that were incredibly wet with really high loads. And again, there were mild bloom years. It's definitely the spring that makes the difference, spring and early summer that drives it. We're starting to see data that suggests that the July matters more, which is not too surprising because if the bloom is growing at that time, then having a pulse of new phosphorus in makes a difference. And I'll note also temperature does matter, but that's for the growth. Phosphorus determines how big a bloom is, but the temperature can determine when it starts and when it ends. Microcystis, that's MAR with the red line, starts growing at about 15, 16 C and thrives well at about 20, 25 or so above there. But some of these others also do well in a similar environment. Lake Erie had a bloom of one of the Dolichospermums, formerly known as Anabana, in late summer, and it can tolerate cooler water temperatures. That's possible. But Microcystis, the most common, is definitely a summer bloom former. We have been working on transport, a 3D model. As I note, the colonies float and sink. They actually literally come up to the surface, often in the day, and sink. And for those who are not familiar with the process, it's fairly kind of simple, rather clever, which is not surprising, a 3 billion-year-old organism. But in the daytime with sunlight, it produces sugars, carbohydrates, and effectively carbo loads, gets really heavy and sinks, becomes dense. That then gets it down to the bottom where there's more nutrients available. It then starts, it's not photosynthesizing and uses CO2, ends out in these gas vesicles, then makes it lighter and it comes back up to the surface. So it provides a competitive advantage in the daytime. It's actually blocking the light from competitors, and it's also tapping nutrients. So we've added that into a model, that floating sinking process, and combine it with the 2D model. So example here is when you add in the wind for mixing, this is a time series. And when the winds are calmer, you tend to get these more intense patterns, and they also vary through the day. And we can put those together. And I would note, this is pretty much characteristic of the scum formers in all lakes. If you're mild winds below about 5 knots or so, you typically will see scums form. If you're up above 10, 12 knots, you will rarely see scum. The wind's mixing is strong enough to disrupt it. And we've been able to generate this, put this together with a model, and actually show the transport. In this case, from this past year, we have the wind here, and I'll note it's spreading up. And the calmest wind, which was right in here, we had the most intense bloom. And then as the winds pick up, it's actually, this is the surface concentration, it's diminishing over time. And pick an example here, very strong winds, and we have a lower surface concentration. In the milder winds, it tends to increase again. But we're able to capture the transport as well as either it's being mixed through the water column or the actual surface concentration as well, using the models. Now, I've got a few more minutes here. I'd just like to touch on this kind of around the country, some examples of what we've been able to do with the blooms. A key effort we've had with the ULCI data is the Cyan Project. It's an interagency project led by EPA and involving us from the algorithmic side. NASA's involved from data processing, and USGS from a field data. And the idea is to implement what we've developed across the nation. The ULCIs, this is the resolvable lakes that can be picked up. The data is actually available here at this NASA website. And there's about what we found is about 1,860 lakes. Resolvable ULCI has 300 meter pixels and about 6 days a week of coverage. If we were to go to higher resolution satellites, we are less specific to cyanobacteria and less frequent data, although we are looking into that because there's an awful lot of other lakes in the country that there's at least concerns of trying to know whether there may be some kind of algal bloom present. This is just a number of lakes we can pick up in each state. From Maryland, out one lake we can pick up to, you can see Florida and Minnesota are among the most, 160 in Florida and 260 in Minnesota as well. This is the coverages. The NASA data is divided into tiles. And so wherever you are, if you're Illinois, for example, you would need perhaps 2 tiles or NASA tiles to pick this up. They are done in geotiffs, so they do look as, they're usable as pictures in their own right, and they also have data, but they're inherently data sets. Geotiff is a really convenient process for that capability. We have been working on severity metrics. This example from Maris, example of looking across the country. And this is the average magnitude in a lake over the season. And I'll note that at least on this example in 2011, that Grand Lake St. Mary's in Ohio and Lake Hancock in Florida had the most intense blooms for the longest period of time. That's not much of a surprise to anyone in either of those states. But you can see there's a number of other lakes in Florida that have extreme blooms, and there's some scattered blooms, lakes that are quite intense in other lakes. And it's possible to look at the trends over time with these. This is just an example from Maris from a paper we have on the concept. And a couple of lakes, one in Ohio and Florida, show they were declining over time. They're getting worse. So this is relative. So these overall are getting worse compared to the other lakes. And then a couple of lakes in Florida that were getting better compared to other lakes. So one effective means of particularly trying to address as you change over time is that, key management question is which lakes do we need to be concerned about? And it's often not necessarily the worst ones, but it's the ones that are changing. And this allows us to look at that, and I can emphasize again the time machine concept with the satellite data as well. Just as a final, I've got just a couple of minutes, I think, kind of a quick run around the country just to show some examples. Lake Pontchartrain, which is a estuary, and it's low salinity, but when the Mississippi is in high flow, they have to run water out the Bonnie Carey spillway into Lake Pontchartrain, and freshwater came out and put freshwater blooms all along the Mississippi coast up here. This is 2 different dates, a few days, about a week apart. And this is area that's normally saltwater. And so we had a freshwater bloom in what would be a saltwater environment. And it was actually producing toxins on the beach. They had beach closures in that area. So the freshwater can be a problem in that case. In Florida, Lake Okeechobee has a lot of issues, and it has closed beaches because of the freshwater blowing out into the coast. Big example was in 2016, significant bloom here, and the water level was so high at the time that the Army Corps had to relieve the pressure on the dike that surrounds the lake, and water went out to St. Lucie. And this is what it looked like in the coastal marinas. This bluish is actually one of the pigments. It's kind of like when the leaves change in the fall, the chlorophyll breaks down, leaving all the other colors. Well, this is the cyan in cyanobacteria, the phycocyanin. But this is all toxic bloom in a coastal marina. In some cases, blooms are showing up later. Lake Champlain actually saw an intense bloom in the first week in October. This is up in Vermont. That's a bit unusual to be that late in the season. But you can see up in Missisquoi Bay and also St. Albans Bay, there's blooms going on. And examples in California, Oregon, California coast, Klamath Lake, Kopko and Iron Gate Lake, there's intense blooms coming through here. This is actually an example of a bloom from up in the Kopko Iron Gate area. And it's possible to identify the frequency. This is work we've been doing with the California Water Board on when these blooms occurred. And these, you can see, these are very small lakes. These are the individual pixels, but we're able to pick up, even at this scale, what's going on in these lakes. And I think just the last one is Lake Utah, which has had some significant problems in 2016. That had not occurred previously and happened again in 2017. Dolichospermum is the case. And you can see where it started, and in this case, Provo Bay, and spread over the lake overall. Some of the lake is also used for watering for crops, and so there's concern also about not using it and to spread any toxins on crops. And so in this case, all of this started in the Great Lakes with Lake Erie. So we're able to look at, monitor, and develop this stuff across the country based on what we've been able to do in the Great Lakes. And I think that wraps it up. So thank you very much.
Great. Thank you very much, Dr. Stumpf. And we're going to be hearing from you during the Q&A at the end. So thanks again. Okay. So hopefully you are seeing my screen now. Our second talk is by Dr. Jessica Ward. Dr. Ward graduated with a PhD in ecology and evolutionary biology from the University of Toronto in 2009. She completed postdoctoral work at Tulane University and the University of Minnesota, and served as a research professor in the aquatic toxicology lab at St. Cloud State University. She is currently an associate professor of biology at Ball State University. Current research in her lab is focused on 3 major research themes: embryonic learning, social learning, cognition, and developmental plasticity in oviparous fish, sensory ecology and the function and evolution of animal communication systems, and anthropogenic effects on animal behavior, With respect to the latter, members of the lab have variously investigated how chemical contaminants, land use changes, and invasive introductions modify the behavior of organisms, primarily fish, and the associated ecological and evolutionary implications of these changes, including population and community structure, hybridization, and the production or collapse of species boundaries and altered selection of traits. So once again, don't forget to put your questions in the Q&A panel, and we'll address them following Dr. Ward's presentation. And with that, I'll turn it over to you.
Okay, great. Okay, everybody can see that? Yep, good. Okay, so thanks so much for inviting me here today. Today I'm going to talk about a little bit of preliminary work we were doing, looking at the effects of some cyanobacterial neurotoxins on the behavior of fish in predator-prey contexts. So I'm going to begin today with the idea that ecological communities are regulated by a whole host of biotic interactions, and these can occur both within species and between species, and the outcomes of these interactions are influenced by information that is obtained from the environment. But the information that is used by individuals to make important fitness decisions can also be affected by the environment. And this is particularly true for aquatic organisms that are subject to a variety of anthropogenic stressors, such as aquatic contaminants or warming water temperatures, or some combination of multiple stressors. So a major focus of the research that we do in our lab is to try to understand the ecological and evolutionary consequences of anthropogenic environmental change on aquatic biota. And specifically, we're really interested in how induced changes in the behavior of organisms affects their survival or reproduction. So one area that we do a lot of work in this area is in regard to how aquatic contaminants modulate the behavior of affected organisms. And we like to see how these changes might alter the outcomes of community interactions. Now, among these interactions, predator-prey interactions in particular have long been recognized as being particularly important regulators of community function. And this is because these top-down predatory forces are really important drivers of how energy moves throughout a system, and it can influence the composition of communities and influence competitive interactions among species as well. So for example, whether a predator successfully attacks and consumes a prey organism is going to depend on its perceptive ability. A predator has to be able to detect a stimulus in the environment. It has to be able to recognize that stimulus as being a prey item and an appropriate something to eat. It then has to be able to physically pursue, subdue, and consume that prey item. And the success with which all of this can happen is going to be affected by the physical properties of the predator and prey and the environmental context in which this interaction takes place. And a variety of anthropogenic activities have been shown to alter the environmental conditions in ways that interfere with predator sensory response pathways. And this ultimately results in a limited ability for the predator in finding and consuming prey. So for example, in both freshwater and marine systems, the reductions in water clarity due to eutrophication and the increased presence of cyanobacteria have been shown to degrade the transmission of visual stimuli. And what this does is result in a reduction in visual detection thresholds, which translates into a reduction in the overall foraging success of fish. These changes can also translate into other community level effects, like shifts in prey selectivity and altered competitive interactions. Other changes in environmental conditions induce deficits in the sensory processing part of this pathway by interfering with nervous system activity. So probably one of the most dramatic examples of this is the effect of elevated CO2 on GABA-mediated olfactory predator recognition in fish. So under high conditions of CO2, GABA-A receptors become depolarizing and excitatory, and this results in the expression of abnormal fish behaviors. So larval damselfish, for example, will actually show a preference for the chemical odors of a predator. So they move towards a predator, which is maladaptive behavior. Many cyanobacterial algal species also secrete a host of metabolic byproducts that can affect the internal physiological function of the organisms. Some of these metabolites are neurotoxic, and they can have marked effects on both cognitive function and then locomotor or motor function as well. So for example, many cyanobacteria surveyed to date produce a neurotoxic metabolite called beta-N-methylaminoalanine, or BMAA. And this is a toxin that's of significant interest because it is also a risk factor for neurodegenerative diseases in humans. BMAA destroys neurons, and there's associations, controversial associations, but associations nonetheless between populations of humans living near affected waterways, or those who consume a lot of foods that are contaminated with BMAA, and various neurodegenerative diseases like ALS, inducing Parkinson's-like symptoms, dementia, and Alzheimer's. Now, animals that are exposed to BMAA also exhibit behavioral, locomotor, and cognitive deficits. So for example, rats that are treated with BMAA during development show strong locomotor abnormalities. In open field tests, pups that were injected with BMAA were found to be hyperactive. So they were mobile for a significantly longer period of time than control pups, as seen in the graph on the top. But they fell over a lot, and they had a very unsteady gait, and they showed marked locomotor abnormalities. Other studies have shown adverse effects of BMAA on the cognitive side of things. So cognition, learning, memory, social behavior, things of that nature. So for example, in a spatial learning task in which rats were tested and retested on 5 subsequent days for their ability to find food in a radial maze, the control animals, which in this graph are shown by the solid lines, showed a reduced number of errors over time, meaning that they entered into empty arms of the maze less frequently on day 5 compared to day 1, which is kind of what you would expect with a normal learning curve. So they got better at this task over time with experience. But by contrast, animals exposed to BMAA did not. So this impacted their ability to learn. So we were given a little bit of money to look into the question about whether BMAA might alter the performance of early life stage fish in the context of predator-prey interaction. So both when catching prey or avoiding predators, either because it impaired sensory and processing systems or because it induced locomotor deficits or abnormalities in behavioral responses. Now, fish will start to forage for live prey items on about the 4th day after hatching. Prey capture is initiated when a potential prey item comes into sight, it's recognized. The larva will orient its body towards that object and begin to move towards it. When it's within distance of the prey, it'll suck that prey into its mouth. So prey tracking requires a high degree of spatial coordination because prey are, of course, moving objects. So this is going to require a larval fish to be able to regularly update the position of the prey relative to its body while it's moving towards it, recalibrate its body appropriately at multiple points throughout this sequence. And fish perform a number of different specific behavioral maneuvers in order to help them maintain this tracking position while moving forward during hunting behaviors. So for this assay, we can measure We can measure things such as the distance to the prey at the time that the hunting sequence is initiated. We can measure various kinematic aspects of the precision of prey tracking during approach. Now, on the flip side, larval fish are often prey items themselves. So during a predator attack, larval fish will often use an innate evasive maneuver called a sea start to evade predators. So this is a different sequence of chained locomotor behaviors from prey strikes, where upon detecting a predatory stimulus, the fish bends its body into this C shape and then burst swims away at a 90-degree angle. And again, though, various aspects of this response can be measured kinematically, such as the latency to respond to a simulated predator, the degree of maximal body bend, the velocity of the escape, etc. Okay, so for this study, we use fathead minnows as our model species. In the lab, we collected eggs from breeding pairs of minnows. We exposed them to a range of concentrations of BMAA for 21 days, and then we assessed their performance on day 21. To assess prey strike performance in larval fish, we put our focal fish in a testing arena, and we introduced live prey items into that arena. In this case, it was 2 to 5 brine shrimp. And these are introduced through a channel that you can see in the lower right-hand side of the picture there. And we used high-speed cameras to film the fish during these prey strike events, and we tracked larval position at 50-frame intervals through these events from the moment of detection or recognition of that prey item through the capture. And we also recorded the frequencies of specific behavioral elements used during the prey tracking, such as J-turns or tail beat cycles. So here's what that response looks like. So in this video here, the larvae is going to be going after the brine shrimp that is in the mid left-hand side of the screen. Okay, and there you got him. Okay, so when we did this, we found that although there appears to be something of a dose-dependent trend, statistically, subjects in all of our exposure treatments initiated that hunting sequence from a similar distance from the prey. So you can see that in the graph on the left. They also demonstrated similar ability to track that prey during hunting episodes. And that's in evidenced by the relatively straight lines that you see on the graph on the right here. So if a larval fish was having a hard time precisely tracking that prey item during approach, then we might expect there to be quite a bit more swing in these lines, but they are fairly straight. So they did a pretty good job of that. And sort of consistent with this, overall, individuals in all of our exposure groups showed similar frequencies of behavioral tracking maneuvers. They behaved in a similar manner, with the single exception that individuals in our highest BMAA exposure group performed somewhat fewer slow swims. Okay, so our takeaway from this is that early developmental exposure during embryogenesis and during the early developmental larval stages really doesn't seem to affect prey strike efficiency or success in any very pronounced way. Now, to assess larval escape performance from predators, we induced a C-start startle response using a trigger-activated vibrational stimulus. So that response looks like this. I'm gonna stop that video. And again, we filmed this with a high-speed camera at about 1,000 frames per second, and we measured various aspects of that response. So we look at the latency to respond to a simulated predator, the degree of maximal body curvature that is expressed, so the expression of that C-shape escape maneuver, the angle of displacement of the head at 10 frame intervals from the frame of stimulus detection through completion of the escape swim. And really that's to see how the expression of this behavior changes over time. Okay, so when we did this, we found a non-significant but dose-dependent trend among our exposure groups, suggesting an increased latency to respond to the predator stimulus. So compared to control individuals, for example, larvae in our highest BMAA exposure group had an average reaction time to the stimulus that was about 63% longer. Individuals in all of our treatments, though, showed an overall similar trajectory of escape once they got started. But we did see another dose-dependent trend in the angular displacement of the head over that first 10 milliseconds of the response. And so that's what you can see in the little inset graph on the bottom right there. Individuals in our higher treatment groups showed a smaller angular displacement of the head within the first 10 milliseconds of response. So what this really means in practical terms is that the control fish are moving faster and they're moving further in that immediate period after detection of a potential predator. We also saw some dose-dependent trends in the extent of the maximal degree of lateral body bend. So this is the physical expression of that C shape with our BMAA exposed individuals, especially those in the higher groups, showing weaker body curvature. So they're not fully expressing this response. Individuals in the higher treatments also took overall a longer time to execute these escape maneuvers, with the total duration of response in our highest exposure group, for example, being about 30% longer than those of the control fish. So from this, we can take away that it would seem that even low dose exposure to some of these algal byproducts during embryogenesis, during these early developmental periods in the earliest life stages, is enough to induce performance-based differences in behaviors that are really critical to organismal fitness. Although the trends seem somewhat stronger in the context of predator evasion, where we see treatment-associated lags in response times to potential predators, other impairments such as longer durations of executing responses. So the question is, does this matter? Do these kinematic changes in performance or performance-based changes, really translate up into issues for populations and communities? So ultimately, we want to know whether these changes in individual behavior might translate into vulnerabilities for actual populations. And this requires measures of fitness. At least some, we are not all the way through this yet, but at least some of the data that we have collected so far suggests that it could. So we also ran some similar experiments with another neurotoxic cyanotoxin, Daba, which is an isomer of BMAA. It is found at similar concentrations in waterways, often in concert with BMAA. Now, in PreTracking trials that were conducted in very much the same way as those I just described, we saw similar trends to what we saw with BMAA. Subjects in all treatments demonstrated fairly similar and robust tracking precision during hunting episodes, again, evidenced by these relatively straight lines. But we also ran some timed foraging trials. And in these trials, these 21-day-old larval fish that had been exposed to algal byproducts during embryogenesis and early development showed significantly reduced foraging success compared to the control fish. Now, everything we've done really has shown that, or suggested that our exposed and non-exposed larvae seem to track prey pretty well. They don't seem to have a problem keeping up with them. So one possibility is that exposed and non-exposed fish are really differing in the probability with which they are detecting local prey in the area. And this is something that we're really just starting to think about, and we haven't gotten all the way through it yet. But when we looked at this a little bit, we found that exposed fish initiated prey strikes from within a smaller spatial region that was anterior to the head compared to controls. So in this inset here, the shaded regions there represent hypothesized geometries of the reactive perceptive field. So that's really the area of space in which a fish responds to a prey item. And those are constructed based on group averages for the initial angle of orientation to the prey at detection and the distance from the prey. So one potential hypothesis for further exploration then is that we see these reductions in foraging success in exposed individuals, not because they have trouble tracking and catching prey, but because they are less likely to detect them in the first place. So performance-based changes in perception and response could have the potential to alter species interactions. This could translate into reduced fitness in the field and reduce larval recruitment to adult stocks. This is important because it can result in declines in native populations and alter community function. And so data like this then are helpful for informing population models that are aimed at predicting the impacts of human-induced environmental change on aquatic populations and communities, which is really our end goal here. Now, in addition, the increased risk of predation mortality that these fish potentially face due to this early developmental exposure has the potential to accelerate the rate of contaminant transfer through the food chain. As I mentioned at the beginning of this talk, BMAA is a fairly controversial risk factor for neurodegenerative diseases in humans. Consumption of foods that are contaminated with BMAA have been putatively associated with ALS and Parkinson's disease and Alzheimer's disease and other neurodegenerative and neurocognitive ailments. So knowledge about the effects of exposure, both on individual species, but then also on the interactions among species that are going to be important to accurately predicting population and community level outcomes is important. And these can also help inform human risk assessments as well. And so that is why we do the things we do. And so with that, I'm going to thank the members of my lab, especially those who were in the lab a few years ago at the time when we were undertaking these studies. Gina Lamko was a master's student who led a lot of this work. Ryan Seymour, Ali Swank, Autumn Oxshire, and Hannah Mullinax were undergrads who did a lot of the daily and hard work of exposing eggs and larvae for months upon end. And I'll thank the funding sources as well. The Indiana Water Resources Research Center was really instrumental in helping us look at these preliminary data, as well as the Indiana Academy of Science. Thank you.
Thank you, Dr. Ward. Really appreciate it. And we are going to spend the remainder of our time with Q&A. Okay, so I want to thank everyone for participating in the Algal Bloom Action Team's webinar series. A special thanks to our presenters, Dr. Stumpf and Dr. Ward, for sharing their research with us. And also thank you to Ann Nardi and her team at the North Central Region Water Network for hosting the Zoom webinar and coordinating all the technical aspects of the webinar. We certainly couldn't do it without her. So as we move into the Q&A session of the webinar, I'd also like to invite all of you to attend our third annual research symposium being held virtually on January 5th and 6th. You will be able to find registration to these events on our Algal Bloom Action Team's website. And I'll drop that in the chat once again, but it has shown up in the chat several times so far. So following the Q&A, please complete the survey at the link that I will also provide in the chat. We appreciate any comments and feedback on this or any of the series. So now we will begin the Q&A session of the webinar. We will draw initial questions and comments from those submitted in the chat during the presentations, and we will do our best to get to all of them. I will start with a question that has that was upvoted, and I believe this one is going to be for Dr. Stumpf. Can you say anything about predictive modeling in riverine systems and what additional factors there may be?
Rivers are hard. They don't stay still. There has been some effort on that in Ohio River, I can think of particularly, and the indications are it depends very much on the timing of the rainfall. That's kind of where that's going. Like wet followed by extremely dry so that the flow slows down substantially. And that's probably a direction that needs to be looked at. They tend to be really episodic when they're not tied to an upstream water body reservoir. But there's a little bit of work going on, but that's a hard one because they tend to be any bloom that occurs every couple of years is much harder to try to sort out. But anyway, some of the work is on the timing of nutrient combined with slowed hydrology.
Great, thank you. Additional question. How do you measure dissolved reactive P?
I don't measure it myself. I work with in Lake Erie with Heidelberg University. They have an extraordinary monitoring program where they have automatic samplers that collect data up to 3 times a day. Standard orthophosphate measurement dissolved is used. I can easily get references. You can check their National Centers for Water Quality Research for that, but it's a pretty much standard method for it. In other areas, phosphorus, I mean, similar method, and it's just the frequency of sampling that's used. Key thing in Lake Erie is the fact that we have the daily data means we have really accurate load numbers. In other areas, it's, you know, you have to develop curves to relate the phosphorus concentration to discharge, and that is an important factor because the concentration does change with the discharge in a water body. You can't just measure it once or twice and assume that that will represent the concentration over a whole season.
Thank you. Is there any correlation with total nitrogen levels and cyanobacteria blooms?
All algae needs both phosphorus and nitrogen. And many water bodies in this country, there's more than enough nitrogen to support a bloom. So they tend to be phosphorus limited. But having said that, there's also a number where there's a legacy phosphorus load that's in the bottom of the lake. And in those cases, it's what we would call an internal load. So the phosphorus comes up from the bottom, gets resuspended each year, reused, and then there is a limitation on nitrogen in those lakes. So for any lake, you do have to pay attention to how that works as to which is the driving factor. Nitrogen leaks out of the system, by the way. Phosphorus will stay, potentially. Now, it can settle out or flow out in other ways, but nitrogen, through a process called denitrification, actually turns into nitrogen gas over time and literally leaves. But there's so much nitrogen typically in runoff that it's not usually the key factor. And there are nuances. The type of blooms you get do vary. There are nitrogen fixing cyanos as well. That, by the way, there's quite a discussion in the community, the scientific community, about the role of nitrogen limitation versus phosphorus limitation.
Okay, thank you. I'm going to give Dr. Stumpf one more question here, and then I'm going to go to Dr. Ward with a few questions. Do the toxins produced by certain algal blooms degrade in the environment? If so, what is the time frame?
Yes, they do. It's generally days, maybe to weeks in some cases in the water. There's some evidence that they can persist longer in the sediments on the bottom, but generally after a bloom goes, there will be some dissolve. And if it's dissolved, that means it's no longer in a cell and it's persisting, but that typically doesn't last for a long period of time in the water. But it can be days to maybe a few weeks, but it keeps degrading over time. I can't give you a half-life, but I mean, we're not talking weeks for a half-life. We're talking on the order of days, I would say, for typical water body in the water column.
Okay, so this is for Dr. Ward, as I mentioned. How do the BMAA concentrations you used in your trials compare to the typical environmental concentrations?
They're actually a little bit lower. So concentrations range widely in the environment. They've been measured from anywhere from about 110 nanograms per liter to about 25 micrograms per liter. And so, so we were looking at concentrations that were in the nanogram range from about 5 to 625 nanograms per liter. To speak to the previous question, we did look at what we could recover in our jars at various points. And for those very low concentrations, we had trouble detecting them. They were below detection limits, within 24 hours. And the higher concentrations were not. So they certainly do degrade within the environment in a fairly short period of time, but they can be persistent. But yeah, so our concentrations were within environmental concentrations, but actually on the lower end.
Okay, so this is also for Dr. Ward. Was there a certain number of trials done per level of exposure in each organism in order to get an average?
Yeah, so our I think for the pre-trials, our sample sizes were in high 30s to low 50s per exposure concentration for each one of those trials. For the sea start predator evasion assays, they were a little bit lower. They were around 20 individuals, I believe, per trial. We also did just some early metrics of hatching and growth and whatnot, where the sample sizes were a little bit higher. But
okay, I've got one, I believe this is for Dr. Stumpf. Does the DRP input later in the summer cause less of a bloom because there is more macrophyte development? And I'm sorry, Dr. Stumpf, you got yourself on mute
I'm
right
muted.
now.
Okay.
There we go.
There are lakes where there is competition between the phytoplankton and macrophytes. If you have developed a healthy macrophyte community, they will do very well on competing against the phytoplankton. So I'm speaking cyanobacteria among the whole range. They will compete very well that way. And that depends on what drives those. To answer the question, you're going to have to go, well, how do you get a healthy macrophyte community in that lake in the summer? And that will vary. Some lakes, changes in water level. If the water level is deep and there's a bit of tannin in the water, you might have very little macrophyte growth, in which case the cyanos will win. If you have little shallower, a little clearer water and the macrophytes grow rapidly, then they will tend to win. They will pick it up. It's very scientific answer. It's all what ifs and conditions and all that sort of thing.
Well, thank you. I think we've got time for maybe one more question. I'm going to go to, let's see. This is for Dr. Ward. Do you have any thoughts about if all individuals in a community are exposed to similar concentrations of toxins, wouldn't one expect the overall effects would be reduced because they are all affecting all individuals, thus someone somewhat equalizing the playing field?
Yeah, that's a great question, and one that we've considered in other contexts. So certainly you would expect that predators and prey would be equally exposed to these, and you know, potentially. So for example, our larval fish that show deficits in predator evasion ability, would the predators also have deficits in being able to catch those prey, therefore somewhat mitigating these effects on prey species and in the increase in predation mortality. We haven't done those experiments with algal neurotoxins, but with other contaminants where we've looked at this, those early vulnerable life stages, the larvae, tend to be more strongly affected than the predators themselves. So exposing a predator and a prey, for example, larval prey, exposing a predator will mitigate some of that predation pressure on prey, but not all of it. So you still have an increase in predation mortality in exposed prey individuals relative to controls, even when the predators are also exposed. So it levels the playing field a little bit, but it doesn't equalize it.
Fantastic. Well, we are up to our allotted time, so I just want to thank again both Dr. Stumpf and Dr. Ward for their presentations today. And thank you all for the questions that you've put forward. We weren't able to get to all of them, so you can see on your screen their emails. If you want to follow up with them, you are welcome to do so. So thank you very much for joining us today.